We'll start with the slides. We've got one model on this test. It's the female ovary model. It'll be in back, quick and easy. We'll get some slides up here, and then you'll bust into the exam. First question on the exam is, recessive genes are usually expressed in the blank condition. Anybody know? Terminology is a lot of what genetics is going to be, plus some Punnett squares on this exam. But recessive genes are usually expressed in the homozygous condition, where it's little r, or little t. That's the first question on the test. Well, I'll get that correct, except for Rachel. So we'll tell Rachel what that question is before she leaves. The slides that are going to be up here, possibilities off of the microscope slides, your last ones of the year. Identify probably the easiest question of the year after you've been through all the slides. We started with slides back in histology. I don't know where this year's gone. It's gone super fast. What are these cells? Sperm. How many chromosomes does sperm have?
23.
23. How many autosomes does sperm have?
22.
22. And one sex chromosome. About half the sperm have an X. About half the sperm have a Y sex chromosome. Organ. Testes. Testes. Structure.
Seminiferous tubules.
Seminiferous tubules. Thank you, Heather. You guys remember this class? No, I haven't seen you for a week. I'm like, we didn't do much over holiday weekend. That's all right. I told you to take it off. Seminiferous tubules. Cell types. Interstitial cells. So if you're writing these down, these are all fair game questions. I'm going to make them up sometime this weekend. These are interstitial cells over here. And they release what? Testosterone. Very good. Testosterone. This mother cell would be called what? And whether do I use this on the slide or it's on the exam, I just took the final this morning, so these terms are going to be on the exam, there's no doubt. This mother cell is called a blank. Spermatogonia. Spermatogonia. So I hope as you're taking notes this morning and I go through these, and as usual, if I'm talking about it, it's on the test. You're at least jotting the terms down if you're not familiar with them yet. Spermatogonia has how many chromosomes?
46.
46. 46. Now, as it goes through the process of spermatogenesis, it will form a primary cell and eventually a secondary cell. The secondary spermatocyte has how many chromosomes? 23. So I would probably write out spermatogonia 46, primary spermatocyte 46, secondary spermatocyte 23. Eventually we have these cells, which are called what? Sperm or spermatids, either one is fine. Chromosome number? 23. 23. Are they mature? No. A lot on that slide is not only relevant histologically, there's also a lot of these cells and terms that are found in the multiple choice questions on the exam as well. So make sure you're very comfortable with these cells and what they look like. Organ. Uterus. Days of the uterine cycle. Days of the uterine cycle.
Oh,
5 to 14. Days of the uterine cycle. 14 to 28-ish. So that puts the menstrual at 0 to 5. What hormone is elevating rapidly during this phase?
Progesterone.
Progesterone. Would the corpus luteum be present at this phase? No. Here?
Yes.
Yes. Here?
No, not yet.
No, it's not formed until ovulation. And so we are in day 5 to 14, we have not ovulated yet. A lot of good questions off the uterus slide. Organ, ovary, cell type, follicle cells, very good, follicle cell. Also, I would accept granulosa cells, some people have asked about that. Structure, you're right on it, antrum, antrum, a fluid-filled capsule that will pop during ovulation. Structure, a glycoprotein coating or protective covering.
Zona
Zona pellucida. Very good. Approximate day of the ovarian cycle. 12. Who said that? Perfect. 12. Oh, yeah, we're written down already. Excellent. That's about 12. So if you're envisioning hormones, and I see 4 that are possible here, which one would be elevating rapidly? LH, I heard it. LH, because LH causes blank. Ovulation. LH is rising rapidly. Do we see any progesterone here? Yeah, no, not really. At its lowest levels, because it doesn't start creeping up until day 14. It'll start to creep up slowly. Good. Those are the slots. All right, 3 sections of this. We're going to do them backwards for a reason. There are 3 chapters, if you will, on this final exam. About 70 questions, all right? About 45-ish, multiple choice, and then matching and Punnett squares to finish it out. It's fair. It didn't take me long to take it today. I think it's fair. I want to make sure I cover some terms that you need to know as we go through this, but I wanted to start, I told you I'd pretty much give you the endocrine. So we wanted to look at endocrine first. So we're just put down endocrine chapter. I'm just gonna basically give you these questions, and so we can move on and make this a reproductive and genetics test, which I prefer. So looking up here, certainly we need to know the correlation between the hypothalamus and the pituitary in the brain. Are we clear on that? The reason we have to know that is because that's where GnRH and FSH and LH are released. And so know the hypothalamus and pituitary arrangement that's happening up here in the brain. There is a question on the thymus and a hormone called thymosin. And so I've asked you kind of to put together a chart, if you will, of hormones and where they're released from, their target, and what they do with their target. Anybody know what thymosin does when it's released? Causes T cell maturation. That's back from the immune system. Thymosin, TTT. Thymosin causes T cell maturation. The 3 T's of the thymus, all right? So you'll see a question about the thymus gland releases a hormone that causes, now you know, release of thymosin, T cell maturation. Other hormones that you should be familiar with, in addition to all the reproductive hormones, you have to know the progesterone, the estrogen, the LH, the FSH, the GnRH, the testosterone. You have to know all those because that's reproductive. But isolated with the hormonal system, you need to know insulin, thymosin, oxytocin, aldosterone, PTH, and thyroxine. Those are hormones. It's a matching section. I have it here on my cheat sheet. I just put the hormone on one side and I give the effect on the other, all right, or the target cell or the release point. You got those 5? Can you match them up? All right, good. In addition to all the reproductive hormones. Don't forget about those. Also, HCG is on this test. Does that ring a bell? It's a reproductive hormone. What's it released by? Good. Trophoblastic cells that are the fertilized zygote that's attached to the endometrium in the uterus. That's what pregnancy tests measure, HCG.
It's released from what?
Heather said trophoblastic cells. I didn't use that term at specificity, but they are trophoblastic cells. Those are just the embedded embryo in the endometrium. At that stage, they're considered trophoblastic cells. Trophoblastic cells are eating the endometrial wall away. I didn't use that term, but that's a good one. It's an accurate term. Good, so make sure you can match those up with their either target or function. Mostly it's function. You guys remember what aldosterone does? Sort of. Could you pick that sodium reabsorption in the kidneys? If you saw that, you go, oh yeah, that's right. Okay, good. So that's an easy section. We'll just bang through that. There's one question on second messengers. There's 2 types of hormones in our body. There's protein hormones and there's steroid hormones. You all with me on that? All right, protein hormones or amino acid-based hormones use second messenger system. Second messenger systems are attaching to a receptor on a membrane and causing a reaction inside of the cell. One question on that, and I just gave it to you. Steroid hormones move directly into a cell and cause gene activation. Steroid hormones move directly into a target cell and cause gene activation, transcription, translation. So only 2 questions on those second messenger versus steroid. 1, steroid, 2, proteins. Third endocrine question. These are 3 triggers for hormonal release. 3 triggers for hormonal release. I gave you 4, and I asked you to choose which one of these is not a trigger. So make sure you know humoral, neural, and hormonal are triggers. It means hormones can trigger the release of hormones. Neurons can trigger the release of hormones. Or blood levels, like blood calcium levels, humoral, cause the release of hormones. Enzymes do not. Got it? How easy is this? Unfortunately, I don't do this for genetics as well, but those are all the hormone questions you got. Don't study hormones. You got those? All right, good. There's like 8 questions, and I just gave them all to you. Figure it out. Know them. Don't not study at all, because you still need to know what oxytocin does. Anybody know what oxytocin does? What's that? Boy, yeah, I'm hearing like 18 things. Oxytocin, what I'm thinking oxytocin does, it does cause milk production, all right? So target is breast. But the interesting thing about oxytocin, some of you probably had. You guys ever heard of a medication called Pitocin? Pitocin is given to initiate the birth process or labor, because it looks like oxytocin. They call it Pitocin. It's a drug given to pregnant moms that are ready to have their baby, but for some reason the uterus isn't contracting it out. They'll give them Pitocin, which is oxytocin, and causes contractions of the uterus.
So you're saying it's similar to oxytocin?
Yeah, similar to oxytocin, it's just a drug. It's a drug that acts like oxytocin in our body that initiates labor contractions. Good. Let's go to genetics. Draw a triple line there. So that's all endocrine. That's easy stuff. Now we're working into genetics. Genetics is going to be some terminology. We're also going to do some Punnett squares. Did you guys all do the extra credit? I hope to God you did. That's just easy points, all right? But those questions are going to come back up again on the exam, so make sure you do the extra credit assignment again. Make sure you can do Punnett squares. The last page of the exam, there are 5 Punnett squares, all right? 5 Punnett squares, and I'd ask you the same questions about probability and chance of this parent and this parent having a hemophiliac child or a Tay-Sachs child or a child with albinism. Are you all clear on that? You have to know whether they're dominant disorders, recessive disorders, or sex-linked disorders. Are we clear on that? And so when I say color blindness, you say sex-linked. We're not very good at that yet. We wouldn't win many ball games. All right. So color blindness and hemophilia, those are the sex-linked. And then you have to also break down the dominant disorders that I provided for you in your notes, the recessive, so that you can just go like that and be able to figure out on this way. There's also a lot of terms on here. I started the exam out with recessive, all right? So if recessive means that it's only expressed in the homozygous condition, like little r, what would you say a definition of a dominant gene is?
It can be heterozygous.
What's that?
It can be heterozygous.
Okay, it can be heterozygous and still be expressed?
Mm-hmm.
Okay. Anything else about dominant genes? Are they good? No, doesn't mean good or bad. What it means is, and this is where you have to pull off that terminology sheet off of Moodle, print it out, make sure you understand it. Dominant gene simply means that if that gene is present, it will mask the expression of other genes. That's what it means. It's going to be expressed. Whether it's homozygous dominant or heterozygous dominant, it's going to be expressed. And recessive is only expressed in the homozygous condition. So you'll need to know your terms. Do you know what an allele is? Well, let's just make a list, shall we? Make a list of terms that are on the exam that you'll need to be familiar with. So dominant and recessive I've already talked about. How about an allele? A-L-L-E-L-E. You'll need to know what an allele is. How about a tetrad? So the more of these terms that you know, tetrads are related to meiosis, the better off you're gonna be. Centromere. I'm not saying these are the answers, but these are on the exam. Phenotype and genotype. If I say dark hair, you'll say what?
Phenotype.
Phenotype. If I say long ears, you'll say what?
Phenotype.
Phenotype. If I say big R or little r, you'll say genotype. You know the difference. Also know the term heterozygous. And know the term autosomes. I asked you a very pointed question earlier, which Heather got correctly when I showed you the sperm. I said, how many autosomes? And the answer was 22. There are 22 in the gametes. Oh, another term, gametes. Another term, zygote. Make sure you can, this will make this test so easy if you can define these things. Good. This is called a karyotype. Karyotype. Would you consider this an allele pair? Yes. That's what an allele is. There are 22 allele pairs and one allele pair of sex chromosomes. 22 autosome pairs, one sex chromosome pair. Is this karyotype representative of a male or a female?
Female.
Female, you look at these last two, and because they're both big and tall, X's, that is a female karyotype. If this was a male, like my t-shirt that I made, there'd be a little stub fragment here that would be the Y chromosome. There is one 16 square punted on this test. If you want to do it, do it. If you don't, you don't. Most of you will be done with this test in 40 minutes. And the ones that are here for an hour, or it's because you're working on this one question, which is the Punnett square, and you have to get it right. You don't have to, and I've already given you a hint. If you understand Punnett squares and you look at the answer, you don't even have to actually do the whole square, you can do it in your head. You'll only get that when you start working it out. And when you see it, it's like, why did I do that? Why did I spend all that time doing this? There's your hint. My undergraduate class in genetics 321, the final exam was a Punnett square problem. You see how this one has 64 possibilities? We had 128. Took an hour and 45 minutes to do one Punnett square, and that was the entire final exam. You got it or you didn't get it. Holy cow. Tough, tough. The final exam is at 8 a.m. on Monday for this class. Are we good? Most of you will be out of here by 8:45. You have 2 hours if you need. Time is not a problem. I will post scores Monday afternoon. All you have to do is post your final cumulative column grade. I don't put put grades on Moodle. So I don't find it to be as secure as people like, but, I'll put your final exam score. You'll have your cumulative column, and all you have to do is go 92, go to the syllabus, and you'll see a 92 is an A minus. We good on that? Your scores will include your extra credit. So that final score that I post is your final score with extra credit. And when a final column pops up on Monday at 2 o'clock, that will be your grade and post, period, end of story, all right? Don't miss it by a point. Don't be that student that misses an A minus by a point, all right? Study hard. Get this grade that you need. I don't feel like I'm the bad guy anymore because we had a nice 5-point extra credit project. So if you missed it by 5, you missed it by 6, all right? Let's not do that. Other things to know. 2 types of fetal testing, amniocentesis and chorionic villi sampling. Which one can be used earlier in the pregnancy? Chorionic villi sampling. Good. So now, triple line, reproductive, majority of the test on that. All that stuff I just gave you, I pretty much gave you. So if you can do it, you can do it. Plus Punnett square. Don't forget, if you just did a triple line and we're into reproductive, don't forget Punnett square problems, because there's like 5 or 6 of them. So they're just, if I said dad's O and mom's AB, you wanna do one? Let's do one. All right, they're just Punnett square problems. What did I say? Dad's O and mom's AB. What's the probability they have a child? Boom, boom, boom. Good. You do it in your head. Did you see what Eli just did? He did it in his head. All right, I can see it too like that because I've been doing it for 27 years. That's what I mean about that 16 square Punnett. Because if you can see it, boom, you don't even have to do it. You just go, well, it has to be that one. Did you see? Here's the probability. What did I say? Probability of an A child. What's the first thing you do? You guys make the biggest mistake. You'll come up and go, I'm not getting this one right. I'm like, what's your genotype? You're like, I don't know. Do the genotypes. Dad is O. There's the genotype. That's the phenotype. Mom is AB. There's the genotype. Yeah, the genotypes, the squares kind of make themselves. I think this is why Eli saw it. Not even having to do the squares. Answer was 50%. What's the chance they have 2 A's in a row? Chance, probability to the nth power, 25. 25 times 25. Good. Bring a phone with a calculator. All right, don't use the internet. Simple genetics problems. Blood typing, sure, we'll do blood typing. Sickle cell, maybe. Hair color, eye color. Figure out the genotypes, and the questions are just like we always do. I would do your extra credit again one more time, because there is a question like Heather posted on the forum yesterday. Males, well, I did it on the final. Daughters. Probability of daughters. So then you have to go back and go, well, which ones are the daughters? They're the XXs. You guys good on that? Okay. Male sister. How many testes? 2. If they both descended, correct? If one didn't descend, what's that disorder called? Cryptorchidism. Cryptorchidism. Very good. Function of the epididymis? immature sperm. Gold's gym. About how long do they live in the epididymis? 20 days. Sperm. How long is the epididymis? This will help you with one question. One question here I read this morning, I kind of chuckled. Which of the following is not true? All right, so you had to go, one of them's clearly true, because we talk about it all the time. It would be like, how many penises do men have? One. All right, I think you guys would all get that right. But it did ask about how long's the epididymis. And I thought, well, I never talked about it. I'll cover it today. It says on the exam that the epididymis is 6 inches long. Would you think that's correct or incorrect? Incorrect, because the epididymis here, I know you can go, hey, it's only like 2 inches long, but the tube itself, if I pulled and stretched that epididymal tube out, you see what I'm doing here? All right, it would go out for feet. 4 to 5 feet is how long the epididymis. I know it's only 2 inches long as it wraps the testes, but it'd be like the intestines. I told you at one time, if I took out my small intestine, could I stretch that up to the parking lot? Yeah, probably could. It's about 25 to 30 feet. It would almost stretch out. Same thing with the epididymis. It's not a 6-inch coiled tube. It is very, very long. So that'll help you out with one
How many
question.
feet was it about?
What's that?
How many feet was that about?
Feet. It's, I don't know, the epididymal structure could probably be stretched out from 6 to 8 feet long. All right, even though you're like, well, it can't be 6 or 8 feet because the testes are only like an inch here, but you know what I'm talking about? Stretching it out. So I don't want you to answer that question. Two questions on the vas or ductus deferens. If you have any questions during the exam, like you studied it at the vas deferens and I put down at the ductus deferens, just come up and ask me. We're old friends now. We've known each other for 9 months. Just come up and ask me. Ductus deferens comes around, enters the pelvic cavity, comes around the bladder, where we get our first additions to semen. Most additions of semen come from what gland? Seminal vesicle. About 60% of seminal fluid come from the seminal vesicle, of which there are 2. How many prostate glands are there? One prostate gland. There's a bulbourethral gland, which is a pre-ejaculate coming out the urethra. How many areas of the urethra are there? Anybody know? It's on the test. 3. Good. 3. Not 4. Good. Dartos, tunic, and cremaster muscles function in maintaining blank. Man, I'm just giving you the question. Dartos, tunic, and cremaster muscles function in maintaining the temperature of the testes. They're muscles that will ascend or descend the testes. Commonly known for shrinkage in cold water.
It's controlled by what? Can you repeat the names? The tunica what?
They're in your notes. Here's the cremaster. And here are the tunica. Does this look familiar? Should. The testes, the seminiferous tubule, the interstitial cells, and all of the structures that I asked about earlier. Which cell, because I'm talking about it's on the exam, which cell at the tip of the pointer creates the blood testes barrier? Oh boy, that's on the test. The sustentacular cells. These are all in your notes, believe it or not. Sustentacular cells, create the blood testes barrier. That's why we haven't ever been able to really create a male birth control pill, because of that little cell that doesn't allow chemicals to get to the sperm. This color, somebody tell me what color that is. That's exactly the color I don't see.
Yeah.
Is that yellow?
Yeah.
Wow. That's the sustentacular cell that creates the blood testes barrier. You'll need to know this progression, people. It's called spermatogenesis. How many product cells are made in spermatogenesis? How many functional cells are made in spermatogenesis? 4. How many do we start with? 1, the mother cell. So know the mother cell, know the chromosome number as it goes through spermatogenesis. Primary spermatocyte, secondary spermatocyte, spermatid, product cells are 4. What are the product cells of oogenesis?
Is that a trick question?
It's could be conceived as a trick question, but it does have an answer. What are the product cells of, if the product cells of spermatogenesis are 4 functional sperm out of 23 with a tail, what are the product cells of oogenesis? Test question.
Polar bodies and.
3 polar bodies and a functional oocyte, right?
Yeah.
There you go. So 4 product cells, but only 1 is the oocyte and considered functional. How many chromosomes do a polar body have?
23.
23. 23. We're still on spermatogenesis here, so make sure you can draw that out and label the chromosome numbers. Makes it easy. One question on this process, which is called spermiogenesis. Spermiogenesis is taking, listen carefully, one cell and streamlining it. One cell and streamlining it. We get rid of the cytoplasm and we form a tail. Spermiogenesis does not make more cells. It takes one cell and makes it a swimmer. Gets rid of the cytoplasm, makes a tail. Spermiogenesis. Think you'll need to know the hormones? Toughest part of this test is this 10 or 15%, both in the male and female system. The rest of them, I swear half of you will be done with 3/4 of the test in 15 minutes. Because it says function of oxytocin. You know thymosin comes from the thymus. All right, you just start, recessive genes do this, or the dominant genes do this. Then you'll get to this. The hormones are the toughest part, both for the male and the female system, there's no doubt about it. I've already reviewed a chart, got them all listed out, done very nicely. What does FSH, LH do in the male system? What does it do in the female system? All right, where does GnRH come from and what's it do? What's it target? But you basically need to know these hormones and what they do in both the male and the female system. And the only way to do it is to study and understand what these hormones do in the male and the female system. All right. LH in the male system does what?
Stimulates testosterone.
Stimulates testosterone release from interstitial cells. LH in the female system does what?
Stimulates ovulation.
Stimulates ovulation. There you go. Ding, ding, ding. So some of those will be easy. They were easy for me this morning. I want them to be easy for you. You'll need to know your hormones, where they come from, target tissue, and what they do at the target tissue. I can't repeat that enough. Make your chart. Make it for both the male and the female system. I told you earlier when we studied the endocrine, no hypothalamus and pituitary. This little axis interaction up here is important. Female. What's the goal of the female reproductive system? Nurture and embryo. Nurture and embryo. All right. How many ovaries? 2. How many fallopian tubes / oviduct / uterine tubes are there? 2. How many uteruses are there? 1. Where's fertilization take place? These are all test questions. In the ampulla of the oviduct. Ampulla of the oviduct. 1 sperm or 20?
Good question.
20. I love this one. What's this remind you of? The model. Good, thank you. The model. Make sure you're comfortable with it. I like it because it actually looks like, you guys have to understand that it appears in this model like these would be developing and going around the outer cortex of the ovary during a cycle. It doesn't happen that way. They develop in the same place. So, but do understand that these are developing. You need to certainly know the corpus luteum. Corpus luteum is on the exam about 5 times because of its significance. It's formed after ovulation. It releases progesterone. It maintains the endometrial wall. Readying it for implantation. And it will degenerate into a corpus albicans if no fertilization and implantation occur. Corpus luteum is 5 times on the step. I like it. What's the chemical cue that will maintain the corpus luteum until the placenta comes in? HCG. That's what HCG is. That's our chemical cue that maintains corpus luteum. Good. How many chromosomes are here? 23. 23. About what day of the cycle are you here? 12-ish? Nothing perfect. I'll take 11, I'll take 13, I'll take 12. What hormone would you expect to see rising at an astronomical rate? LH, because it causes ovulation. Good. If I put a pointer here, what could you tell me about LH and FSH? Can you see it? What day in the cycle are we? 15? Beautiful. 16, 17, we're probably 15 here. 16, 17, 18, 19 here. Tell me about FSH and LH on day 18.
They're inhibited. Yeah.
Plummeting. They're going down dramatically. Can you answer questions like that? Because I will ask you questions like that. On day 18, would you expect to see A, progesterone rising and LH falling? Yes or no? Progesterone rising and LH falling. That's the right answer. That's what you see. You're gonna have to see that. All right? Pretty. There's the ampulla, that's where fertilization takes place. Endometrial wall, vulva. Talked about oxytocin already, nothing about breast tissue, areola, lactiferous ducts, nothing came up on the exam. Oogenesis, oh my God, yes. Just like we did for spermatogenesis, can you pick out a cell and tell me how many chromosomes? This is oogenesis. How many product cells of oogenesis? 4. Are they all functional? Nah, this one's considered the functional one, and 3 polar bodies. But we now know that the polar bodies can be fertilized, but they're still not considered functional gametes. I don't trick you on that. I don't even ask you about that. Too confusing. How about this? Think you need to know this? You have to know this. These are the big ones. These are gonna separate the A's and the B's, right here. Can you put this on a day chart? I can. Put this on a day chart. Right here. It's probably day what?
14.
14. So put it on a day chart so that when I say on day 8, you'll just go like this. And on day 8, LH is maintaining, rising quickly, falling quickly. Maintaining. There's day 8. See how this works? All right, come down here. We just did day 18. Do you remember that? I just showed you day 18. And I asked you 2 questions. And you said that LH is falling, but progesterone is rising quickly. Would that represent this map? Yeah. You have to know the hormones. Not only for the female system where it's more complicated by a long shot, you also have to know it for the male system. 4 major hormones are LH, FSH, estrogens, and progesterone. Somebody asked me on the forum, or no, on an email, if we need to know the difference between the estrogens like estradiol and estrone, and I said, no, never talked about it. All right, but those 4 big ones. There's a nice picture of it. If you're a map person or a drawing person, just like spermatogenesis, there's what the ovogenesis feedback loop looks like. Yep.
Stan, if you don't mind going back to that previous picture, what was a little confusing at step 8, it said estrogen, progesterone, and inhibit, oh, sorry, one right there.
Yep.
That, the arrow is saying it's inhibiting.
Yep.
I'm not sure how that fits into our context.
These are negative feedback mechanisms that will actually inhibit the release of GnRH. So basically it stops releasing LH and FSH while the corpus luteum is present for the next 12 days. We stop, we freeze. Because we're waiting for what?
Baby.
Implantation and pregnancy. And therefore for the next 12 days we don't want anything going on that might cause FSH to be released, because FSH actually causes more follicles to develop. So really from day 14 to day 28, the hypothalamus and the pituitary are inhibited or arrested. We don't do anything. They work in the first 14 days and then stop, take a little break, 2 weeks in a white, then they come back on day 1. All right? That's how that works. That's important. Male system have inhibit?
Yes.
Yeah. But in that context, it's so we don't make 40 million sperm per hour. And then match it together with the phases. And nicely on this, and this is in your lab manual too, they did put the days down here. So again, we said day 18 earlier, and I like day 18. There's a lot going on. LH and FH up or down? Down. Progesterone up or down? Up. Endometrial wall growing or falling off? Growing. Because we're ready for what? Implantation. Everything's right for implantation. If we get implantation here, is the corpus luteum going to degenerate?
No.
It is not. Because this implanted embryo, trophoblastic cells that we started with this morning, release HCG, and will maintain the corpus luteum for about 3 months until the placenta takes over. Those are the different phases. How does sperm move down the ductus deferens? Peristyle. One question on birth control. Let's give it to you. What is the term for the device that's implanted in the uterine wall to prevent implantation? IUD. We didn't talk about it, so I'll just give it to you. covered it all. Are there any questions? Any questions? About 70, take about 45 minutes, 8 o'clock Monday. I'll be online all weekend at the tournament in Salem, but I'll take my iPad this weekend, Salem. I'll be available. All right. Post any questions. It's been a great year. If you won't listen to me on Monday, here's my rule. You'll be too nervous. You want to get this over with. You guys will do fine on this test, but study for it. My goodness, this is a test. If you want an A, you can get an A. You can get an A+ on this test. The highest and maximum points that you can get on this exam is 107, because it's a 5-point raw extra credit, and there are usually 5 107s per year. So go get it. All right. There's no surprises here. Go get it. I hope you get the grade that you want. It's been a great year. I can't believe how fast it's gone. Here's my rule. I'm gonna see you guys in town in a year or two, and I'll remember your face, but I won't remember your name, because I get 300 of you and you get one of me. And I'm always gonna be your A&P teacher, all right? But you're gonna be one of my many students. So come up and introduce yourself, and just remind me of your name and tell me when you had me, and then we can start the conversation, because I don't want you to feel offended that I don't remember who you are. I remember your face and who you are. I just don't remember names very well, because I'm getting old. Fair enough? If there's anything I can do to help you out with your future endeavors, let me know. You know where my office is. Have a good weekend.